And here we go. So again, welcome. This should likely take about an hour, but if you guys have anything that you need to do, hop off, come back in. You're absolutely more than welcome. So we'll get started. So for those of you who don't know me, my name is Rachel Bahouth. I have been the student lead on the Nest Parasite project for the past 2 years. You've probably gotten a lot of emails from me throughout the years. I really appreciate it when you guys respond, but just a little bit more about myself. I'm a biological sciences major with minors in ecology, evolutionary biology, and communications. My favorite parasite is the parasitoid wasps because I think they're just so little and cute to see under the microscope. And my fun fact is that I love to travel, and prior to COVID, I had visited 14 different countries.
Hi, I'm Mahima. It's so great to see you all. I am a junior, majoring in molecular and cell biology with a minor in Spanish. My favorite parasite is the blowfly larvae. I think they're so interesting. They look like tiny little worms. And a fun fact about me is that I am the president of the Yukon Pre-Medical Society and the president of the Yukon Seeds chapter.
Hi, so my name is Caroline Webb, and I am currently a sophomore at UConn. My major is environmental science with a human health concentration, and then I'm also minoring in environmental economics and policy. And I'd have to say that my favorite parasite are the blowflies. Just the color, we'll show pictures later. They're pretty under the microscopes. And a fun fact about me is that in addition to working in the lab, I also am a member of UConn's Division I cross country and track and field team. So pretty busy, but I really enjoy working in the lab. And I'm hoping that you'll get a little bit of a better idea of what we do today.
So of course, there's one more very important member of our crew who could not be here today. Dr. Sarah Canuti is currently driving home from Minnesota, so she's not able to make it, but she is the principal investigator and an assistant professor here at UConn. Her favorite parasite is the bird flea, and her fun fact is that she camped out in an active volcano in the Philippines for 10 days, which is definitely a very fun fact. So Sarah is responsible for starting this study back in 2018. And although we wish she could be here today, she gives all of you her best regards, and she's so glad that you guys could all make it.
So the study, the what and the why, what are we doing? Our goal is to determine what parasites are where and how these parasites affect the eastern bluebirds and tree swallows that we're studying.
So on this slide, we have two maps. On the left side, we have a map of the range of the eastern bluebird. And you can see it's mostly in the eastern side of the US, but kind of goes all the way out until the kind of like the midway point in the country. And then you can also see based on the different colors, the purple is where the eastern bluebirds are year-round. And then a little bit farther north is the breeding season, and then a little bit farther to the west is the winter season. And you can also see in the map, there's little black dots, and each of the dots represent a county from which we receive nests. So it's pretty cool. We can see that there's black dots kind of spanning most of the range of the eastern bluebirds, which is pretty cool that we get such a wide variety of data. And then on the right side, we have an image of the tree swallow range. And tree swallows have a little bit of a wider range in terms of where in the geographical region of the US, but we also do, you can see by the black dots, we do have a wide variety across different states from which we receive nests.
So I'm sure a lot of you guys have been landlords for a while now, but if you are new, the landlord's process, what do you do? What are you responsible for? So we ask that you count the number of eggs, whether they've been hatched or unhatched, nestlings, whether their fledging was successful, unsuccessful. Make note of whether you're feeding mealworms or not, because this is important data for Rachel's project that we'll talk about in a moment. And also, once you're done with this data collection, we ask that you collect the nests from the nest boxes, place them in a Ziploc bag, and then ship them to us to get ready to be processed.
So here are just some quick facts about our study, just some things that we think are super awesome and we would love to share with you guys. So in total, we have 142 landlords that have signed up to be a part of the study. Not all of them have sent in nests, but as of now, it's 142, which is a huge number from where we started in 2018. We have approximately 23 different states represented from the landlords that have signed up, which again, as you saw on the last slide, is awesome because we really gives us a big area of the ranges of both of the host species. And then, as we said, our main host species are eastern bluebirds and tree swallows. And to date, we have received just over 1,000 nests, which is awesome. And we're so excited to finally see a 4-digit number with a comma. We are so happy about that. And as we kind of mentioned already, so far we have been doing the study for 3 years and soon to be 4 with this upcoming season. So 2018, 2019, 2020, and soon to be 2021.
So one of the most commonly asked questions that we get is, where's my 2020 data? And we're hoping that through this presentation, we'll give you a little bit of a better idea of the process of going through the nests and how it can sometimes take such a long time in order for us to be able to give you back data that we feel confident that is as accurate as possible. So here, kind of going over a little bit what the process looks like after you send us our nests. The first thing that we do is that we catalog all the nests that we receive. So I know many of you have been participating in the study for at least a year now. So you'll know that as soon as you receive the nests, we'll send you an email, make sure that we've received the number of nests that you think you sent us. And then after that, we go into the nest dissections themselves. And so this involves kind of taking the nest out of a plastic bag and going through it with a pair of tweezers, pulling apart all of the different branches, all of the pieces of grass, the feces, the dirt, looking through everything, and counting all of the parasites that we see. And then we also collect the commensals. So commensals are non-parasites that are found in the nest. So these will be things like caterpillars or beetles that don't feed directly on the bluebirds themselves, but still are important part of the community within the nest. So we do make note of them. We just don't count them the same way that we do the parasites. And then after we've collected all of these insects, the nest thing that we have to do is to identify the parasites. And we'll elaborate a little bit more on this later, but this is also a really important process in order to make sure that everything that we've collected is actually what we think it is. And then last but not least, once we have all of this data, we can analyze it and start to put it together in order to come up with conclusions about what we've seen. So on the next slide, you'll see a time lapse of a nest dissection. This is only about 15 seconds, but actually going through the nest can take up to like 2 to 4 hours, depending on how dense the nest is or how many parasites that we find. And you can also see in this video, standard lab protocol, we'll be wearing personal protective equipment. So we have the lab coat on, we have the gloves on, and then I also have a dust mask on to protect myself from all of the dust from the nest. And then we just kind of go through all of the material with a pair of tweezers.
So what are some common parasites that we find in your nest? First, we have a picture of a flea, which is not to size. They're actually so tiny that I often get them confused with dirt, which is why the IDing process is so important. And next we have a picture of a blowfly. And as Caroline mentioned, this is her favorite parasite, but I think all of us have a bit of a liking towards this particular parasite because it's so beautiful. It has this really great iridescence, which you can kind of see in this photo, which for me makes it really exciting when I find it. Next, we have some mites. These are actually two different mites. And again, similar to the fleas, they're very tiny, actually smaller than the fleas. So to the naked eye, we would think that these two are the same, maybe just a speck of sand, but actually these are mites and they're actually very different. These are two predator mites, and we also find non-predator mites. They look kind of like little pillow sacks, which we'll show a picture of in the future slide. And here we have a picture of a parasitoid wasp, two parasitoid wasps on top of a blowfly pupa case. And so parasitoid wasps are basically parasitites of a parasite, which is really interesting because it shows that there are layers to the communities of parasites that we're finding within your nests. And last but not least, we have a picture of a bird lice. And don't worry, these are specific to birds. So you touching your nests or us touching your nests won't cause any transfer of any lice, nothing to worry about. They only affect the birds.
So as Karen and Mahima have kind of alluded to, one of the most important steps in the process is the identification of the parasites that we collect out of your nest. And this would not be possible without the help of Dr. Matt Bertone, who has been our collaborator. We are so happy to have him on the project. He has really been very instrumental in teaching us and educating us on this identification process. And so here's an example actually taken from a nest that was sent in by a landlord. And so here you can see mites and lice. So we have the book louse right there in the center. You can see it's obviously a little bit bigger. The body structure is very different. And then right underneath that, you have a parasitic mite species. It's considered a mesostigmata. And then all surrounding that, you can see the dust mites, which are kind of like Mahima alluded to, little white pillowcases. They're extremely small. I really cannot stress that enough. They are often, they are almost like smaller than a grain of sand. But this is obviously under a microscope, under very high magnification. And so you can see how, this process of identification definitely takes a long time because you're looking at so many very microscopic species, and it's really important for us and for the accuracy of our data to make sure that we are identifying everything correctly. And here's another example, again, taken from a nest sent in to us by a landlord for this past 2020 season. And so you can see so many different types of not only parasitic mites and dust mites, but also predatory mites. So this is commonly what we are seeing when we are identifying parasites, and it definitely is labor intensive, but it is really cool to kind of separate everything out, and we are definitely learning a lot as undergraduates at UConn, and we're really able to get a lot out of the identification process. And so now we wanted to kind of test your knowledge. So we know that this picture has a lot going on, but we kind of want to play a little bit of an I Spy. And we were wondering how many predatory mites and parasitic mites you guys can actually count in this photo. We know it might be kind of hard over Zoom and camera quality and all that type of stuff. But this is a sample that we went through in the lab with Matt Bertone. And there are different types of mites in here. So if you guys just want to take a quick second and count out how many you think and want to throw them in the chat or unmute yourself too, we definitely love to see how many you guys think that you can find. So I'll give you guys a second to try and count them. All right, so everybody counted. Everybody have their number in their head for a game of Might I Spy. So the correct answer is 11. So you can definitely see how it definitely can take a while to make sure that you can count all of the correct mite species and make sure that we're all very accurate. which is really important. And so you can obviously see with the blue circles. So you might have seen the 3 main ones in the center, but then the 3 off to the right-hand side might have been a little bit harder for you to see. And that's really where we're very happy to be using microscopes and lots of wonderful lab equipment that make this process a lot easier on everybody involved.
Sorry. So what's next with our study? First and foremost, our biggest priority is to consolidate the 2020 data. And by this, we need to identify the parasites that we're finding, similar to the pictures that we just showed you. An exciting process that's happening is that with the next semester, we will actually be having 6 undergraduates as part of our nest parasite team. So possibly help us get through these nest dissections a little bit faster and get that data to you a little bit faster so we can all be excited to see what kind of parasites are inside your nests. Interestingly, actually, Rachel is currently working on a project with the data that she's been collecting from your nests regarding food supplementation and their effect on parasite abundance. And I just got awarded funding for my own grant where I will be looking at how temperature affects the DNA of eastern bluebirds.
So I think now we'll get into the question and answer. So I'm going to stop sharing my screen. So thank you again to everybody who submitted questions. We got some really good ones, and so we're super happy to answer them for you. So the first question that was submitted to us is, what is the minimum and maximum number of nest sites ideal for your research?
So ideally, there is no maximum number of nests. The minimum, of course, is 1 nest. So to participate, you have to be able to send us at least 1 nest per season. However, due to the limitations of the size of our undergraduate crew, we ask that landlords send a maximum of 50 nests per season. And again, we are happy to have anyone participate. We know that some of you don't have 50 nests per season, and that's totally okay. Again, we're happy with any nest that you are able to send us. And another question that was asked was, why specifically are we looking at bluebirds and tree swallows and not other birds that have a range in the US?
And this is a really awesome question. And actually, Sarah, Dr. Canuti, has a long-term field study site in Minnesota where she's been studying these 2 host species for the past 7 years. And what had happened is landlords and other people with these box nesting birds, the eastern bluebirds and tree swallows, noticed that there was such a large variation in the types of parasites that these 2 birds had, and were wondering if there was a relationship between these parasites and their geographical area. And, you know, in addition to this, luckily, bluebirds and tree swallows are perfect study hosts because they are box nesting birds that have such a large geographical range. And that makes it really awesome for us to study and see how their location impacts their parasites. Another question that we received is, well, why do we care about nest parasites?
So this is actually a really great question. So the developmental stages, or the early stages of a bird's life in particular, just like any other animal species, if you were to look at it, are really influential in determining what they will be like as an adult. And when the birds are young, that's when they're the most likely to be parasitized, which then helps us furthermore analyze what we think they might be like as an adult. And so that's kind of why we care so much about the nest parasites during the breeding season and when the eggs are hatchlings. The next question that we did receive from a few of you multiple times actually is, should you remove house sparrows or other birds from the nest boxes?
And this is actually a pretty complex question. House sparrows are an invasive species in the US. However, they've been here for a really long time. And realistically, we aren't going to be able to get rid of them anytime soon. And so as a study, we don't take a stance on whether or not you should remove house sparrows or other birds from your nest boxes. And we leave it up to the individual landlord to do what they think is best for themselves and their nests. And so another question that we've received is, which parasites harm humans and in what ways can they affect us?
Yeah, and I kind of touched about this during the presentation, but so fortunately, all the parasites that we work with in our study are specific to birds. And so this means that, none of the parasites that we're looking at interacting with can live on us. Although it's possible for some of these parasites to possibly bite us, it's super highly unlikely, but this is why we recommend you wear gloves when handling the nests. Another question we received is, does food supplementation help the birds resist parasites?
So Dr. Canuti has previously studied this at her long-term field site in Minnesota, and she had found that food supplementation does in fact lower parasite abundance in the bluebirds. And so I decided to take this a step further, and using the Nest Parasite Community Science Project as kind of my study system, I am now testing to see if what she found holds true throughout the entire geographical range of the bluebird. And so far, our current analyses have shown that supplementation does in fact lower parasite abundance. However, it is dependent on location and other environmental factors like the year or temperature and other things like that. But I will be working on a manuscript for that, so you might be seeing that in an email sometime soon. Our next question is, when will we be receiving the results of what was found in our nest from the 2020 breeding season?
So I already touched a little bit on this during the presentation. As you saw, it can be a long process, especially when some nests can take up to 4 hours just to dissect them. And then of course you have to go through and ID everything that we found. However, specific to the 2020 breeding season, with COVID-19, it has been a little bit more difficult in order to get into the lab with university protocols, which are obviously there to keep everyone safe, especially if some of us don't live in state. It has been a little bit more difficult. However, we do anticipate getting this data to you within the next few months. And again, we're so happy that all of you are participating in our study, and we really value your participation. And so also next year we're doubling, as Mahima mentioned, we're doubling the size of our crew from the 3 of us now to 6 undergraduates. So we're really excited about that, and the possibility of getting the data to you a little bit quicker next year. And again, just reiterating that we really want to make sure that when we send it to you, it's as accurate as possible. And then another question that we received, which is actually something that when I first started the lab, I was wondering myself, was if you put your nest in the Ziploc bags and 2 weeks later there are still flies alive in the Ziploc bag, like, is the bag not airtight, or what's kind of going on with there?
And this is a really awesome question, and something that I myself wonder, and Caroline wondered as well. So to touch on this, the blowflies, due to their larger size, do happen to be dead when we're processing the nests. And this is true with some of the other larger bugs as well, such as wasps and bees. But, the smaller parasites that we look at, such as the fleas, lice, blowfly larvae, mites, and other smaller bugs, such as the caterpillars and small beetles, they do happen to be alive when we're dissecting the nests. And this is just due to the fact that they're just so small in size that they have enough air inside of the bags to survive the time from when you put them inside the bag to when we open it back up to start dissecting. And actually, we prefer that the the parasites are alive when we're dissecting the nests. And this is to ensure that the DNA has not begun decaying, because there's a lot of valuable information in there. And so in the future, if we do plan on doing further research inside the DNA and understanding more about these parasites, we have that data ready for us when we decide to do that. Another question that we got is, what are the bad parasites and what can we do about them?
So the answer to this question is, might not really be something that you're expecting, but the parasites have been around just as long as the birds have. So unless the birds are in really poor conditions or they're just not doing well to begin with, the parasites are not really as damaging as you might think. Birds need to be exposed to a lot of different things to ensure their survival. And so exposing the young nestlings to parasites when they're younger is typically better, so that they can survive to adulthood and not be as impacted from it later on. It's kind of similar to like kids and allergies. Kids need to be exposed to things when they're young, and then it's not that big of a deal when they're older. It's very similar. The immune system works in kind of a similar way in birds. Our next question, what is the most common parasite that you find?
So this depends on the nest, this depends on the region, but in general, the most common parasite that we find are fleas. And again, depending on the nest, some nests we only find like 1 or 2 fleas, and in other nests we find hundreds. I've gone through multiple nests where I found over 500 fleas in a single nest. Which again kind of shows you how long the process can take in terms of counting every single one of those fleas in order to have accurate numbers and accurate data for our study. And then another question that we've received is there any way landlords can see what parasites were found in their nests?
So the answer to this question is yes. At the end of the breeding season, once we have consolidated our data and analyzed it, the data is sent out to each individual landlord in an email, and it will include a data sheet with the number of parasites that we find in each nest and just other general information that we found was interesting to us that we think will also be interesting for you. And last but not least, we have a question for you. Is there anything that you're interested in hearing more about, or any other questions while we were talking that formed for you, or anything at all that you'd like us to talk about, clear up?
And you can either unmute yourself or put it in the chat, kind of just an open forum now to hear from you guys. So.
I'll ask a question. I thought you were going to actually say it, but you went into the house sparrow answer. Do you have recommendations about cleaning the nest box out between broods? So if we're taking the nest out, do you have any suggestions? Should we like super clean it, or just get the nest out and leave it for the next brood?
Yeah, so I think I can kind of take this. I would say there's no real protocol that we would ask you guys to follow. Just clean it to the best that you can, but try to not use really any harsh chemicals that could possibly be influencing the parasite load of the next brood. So if you're using, like really intensive Clorox and bleach, that's not going to be great for the study results of whatever could come from the next. So like similar to how we ask you guys not really to use insecticide, try to be cautious and mindful. But again, it's kind of up to your discretion. I mean, if it was my nest box, I would probably suggest cleaning it decently, and so that the next brood can have a nice, clean place to live.
And also to address the question in the chat, I just brush out the floor and walls. Is that enough? Yes, that's fine. Again, we don't really have any specific protocol that we ask you to follow. It's just important, as Rachel said, to not use anything that's strong enough that could impact the nest parasite community. So if you guys also have like any other questions, it could be about like us, the study, what we're working on, about the birds themselves, about your role, or any comments, anything that you'd be interested in hearing from us, we're happy to take anything.
So in the chat, what do you guys do with the data regarding the number of hatched versus unhatched eggs? Mahima, do you want to take this one?
Yeah, so this data, aside from immediately writing it down once we get that information from you, this is helpful for us to understand, maybe what type of effect did the parasite have on this bird. And if we notice that, you say that the bird had laid 4 eggs and none of them hatched. And we also noticed that there wasn't a really big abundance of parasites. That makes us wonder, well, is there a correlation between this? And this is patterns that once we start seeing this often, we can start to maybe make conclusions for, or possibly open the doors to more studying and finding more answers.
I'd just like to build off that. I think Mahima answered it so well, but this information is really important for also, long-term study data. So, with that information, if the study goes on for the next 10 years, and you can look back and say, oh, wow, a lot of the nests that we received in 2019 had so many unhatched eggs, or so many nestlings died. What was that? What happened there? What was going on within the ecosystem, within the community, environmental conditions. So, that data is really important it can be applied to a lot of other stuff, more so than just birds and parasites, but also a lot of environmental factors as well.
And then I see another question in the chat. What if the nest is abandoned with eggs before hatching? Would it have parasites? And then would that still be a nest that has information for you? So it is definitely possible that the nest could still have parasites. Again, some of the things that we found are parasitic, but other stuff that we do look at are commensals, or things like caterpillars that don't specifically feed on the birds themselves, but are still noteworthy in order to understand the nest parasite community. And it is still valuable to have that information about a nest that is abandoned. Sometimes it's especially useful. I know that there have been landlords in the past who sent in nests and who might say, oh, a snake that they've watched got to the bird, and that's the reason why the nest was abandoned. So clues like that obviously are important in understanding why, but even without that, it's still useful information for us to have in the study. I don't know if Rachel or Mahima, you want to elaborate?
Yeah, what Caroline said is perfect. The next question that we have is, if we have parasites in bluebird nests, is it likely that we will have the same parasites in other species nests like purple martins or barn swallows? So I would say it is probably likely that if you are having bluebirds or parasites in that nest, that it is likely you will see them in your nests of other species, because if they're in the bluebird nest, that means that they're in your area. They're in your range, they're in your area, they're in your land. So although it might not be exactly the same, it is probably likely that you will be seeing them. Some of these species of parasites are host specific. So some might be specifically for bluebirds, but that's not all of them. So I would say you're likely to still find fleas and dust mites and things of that nature in your other nests as well. So if you guys don't have any other questions, which if you do, keep asking them. We love to answer anything else from you, but that's kind of all that we have prepared for today. So like we said, this is going to be recorded. It's going to be put out. We are going to edit it a little bit. So if you don't want to be in it, that's totally fine. But if you have any more questions, feel free to ask them. And if not, we just thank you so much for coming. And we really value your input, and we'd love to hear some feedback at the end. Thank you. We love your energy as well.
And as always, if anything comes up, you can always shoot us an email and we'd be happy to help you out. Or if there's something that you'd like to talk more about, we're, as you can tell, very excited about this, and we love this project, and we're more than happy to chat more about it.
Hi, Sandy. Hi, Steve. We love you. You guys are our biggest fans. We like when you comment on the Instagram.
We love you guys too.
Thank you.
Thank you. We're not very good on this, so.
No, thank you so much for coming.
You're welcome.